Yeah, let's begin the second presentation. The second speaker is Dr. Rozeita. She is the Deputy Director of the Pest Disease Management Program, Horticultural Research Center, Malaysia Agricultural Research and Development Institute. She has 20 years of experience in pests and disease management of horticultural crops. And the title of her presentation is the status of the bacterial wilt disease of banana in Malaysia. Let's begin. Thank you.
You are not audible.
Hello.
Yeah, this okay.
Yeah, you can hear me. Yeah, all right. Okay. Hi everyone. Assalamualaikum and good afternoon here. First of all, I would like to say thank you to Dr. Lee Chun-Yu from the chairman for this session and for the brief introduction. It is a great honor to be here today with all of you to share my slide on the status of bacterial wilt disease of banana in Malaysia. Before I go further, I would like to take this opportunity to congratulate TFNet and MARDI for the organizing of this virtual workshop for the half day. Yeah. As introduced, I'm Dr. Rozeita from Horticulture Research Center, together with all my colleagues from different research center. And we will be presenting the status of bacterial wilt disease of banana in Malaysia. Yeah. My presentation will cover some introductions, mostly on industry scenario, problem statement, the major banana productions constraint, the status of banana bacterial wilt disease in Malaysia, and number 4, disease management strategy, which include a government initiative and R&D. And lastly, on the conclusions and way forward. Okay, let me begin with some background of Malaysia. I hope everybody know where is Malaysia. Yeah, Malaysia is situated in the Southeast Asia. Yeah, our current population is estimated of over 32 millions. The country is multiracial and multicultural, contains of Malay, Chinese, Indian, and others. We have 13 states and 3 federal territories, and separated by South China Sea into 2 regions, that is Peninsular Malaysia and East Malaysia, or in Sabah and Sarawak, or normally in Borneo Island. And our national language is Bahasa Malaysia, and English is a second language. Yeah. Now we go to industry scenario. From our record, agriculture remains as important sector in Malaysia's economy. Presently, agriculture sector contribute 7.4% to the GDP and provide about 10.5% of total employment in Malaysia. Yeah, this is recent data. In Malaysia, agriculture sector, banana is one of the popular and commercial fruits for local and export market. It is ranked the 3rd most important fruit crops cultivated in Malaysia in terms of total production, and as well as 3rd rank in the fruit export revenue among the other major fruits, yeah, especially watermelons and durians. Okay, next, Still in industry scenario, banana contribute around 21% of total fresh tropical fruits and 14% of the total fruit areas, covering more than 26,000 hectares of land with estimated production volume of more than 330,000 tons. There was a slight increment in terms of banana total productions and hectares from the previous year, which is in line with the Malaysian government initiative in National Agro-Food Plan Policy 2.0, targeting to increase the fruit subsector productions for the next 10 years, starting from 2020 up to 2030. As compared to other short-term crops, for example, papaya, banana is the priority crops in terms of agricultural land use, and this cash crop remains in the top 3 of the highest per capita consumption, which is about 9.3 kilogram per year, or we consume about 25.4 gram daily for 32 million Malaysian. Okay, so having said that, regardless of any crops, of course they have the major constraints that reduce the yield reduction. In general, if you can see that table 1 and table 2, for the past 5 years, the trend of banana planted areas in Malaysia has shown to decline from 34,000 in 2017 to 26,000 in 2021. You can see that in the table 1. Similarly, the trend of banana production from 350,000 to 330,000 in 2021. Despite the downward trend, there was a slight increase in both banana plantation and production in 2021. One of the contributing factors to the deterioration of this production is the major disease infection. So now we go to the status of bacterial wilt disease in Malaysia, as we expected. Banana bacterial wilt disease is one of the major diseases caused by the bacterium Ralstonia syzygy subspecies solanaceae, commonly known as a blood disease, and Ralstonia solanaceae raised to bivalve 1, commonly known as MOCO. But in Malaysia, the blood disease is more pertinent as compared to MOCO. This bacteria was first detected in 2007 in Johor. It's almost 15 years ago, and result in remarkable yield loss and increment in banana importation from the value of $1.7 million in 2007 to $36 million in 2012. That's a huge amount of money due to the losses. Bacterial disease is considered the most damaging disease of banana and has been listed as one of the top invasive alien species in Malaysia. As we know that the disease is easily spread by contaminations of planting material, farm tools, water. Pathogens could survive in the soil for more than 2 years, without any host. If there's host, I think it's more than 2 years. The infection during the outbreak could be up to 100%. Until today, no agrichemical effectively control this notorious disease. This just to show to you that the survey done by Department of Agriculture in 2012 from the data that almost all the banana cultivars in Malaysia has been infected with bacterial disease. Even during that time, pisang boyan was escaped from the infection. However, unfortunately, soon after that, none of banana cultivar were found free from the bacteria. Recent survey done by Department of Agriculture in Malaysia recently in 2012 found that out of 300 hectares, only 14 hectares, or is about 4% of disease infections are caused by bacterial disease. That's the latest disease survey by Department of Agriculture found out just about 4% of disease caused by bacterial disease. This is a very good indications that the bacterial disease infections and spreading have gradually decreased. This is also given an indication that the government, Malaysian government initiative or strategy to contain the spreading of the bacterial disease is almost succeed. So what are the government strategy or initiative to reduce the bacterial disease spread? So here I list down the 10 initiative from the government or regulations or enforcement to reduce the spreading of the disease. One is biosecurity enforcement based on Plant Quarantine Act to introductions of contaminated to protect the introductions of contaminated plant material from outside, and regulation to restrict the movement plant material from disease infected to other location. Number 3, regulation to destroy or mutilate infected plant banana material from farmers' field. Number 4 is application of sanitation in farmers' field, and then regulation, monitoring and surveillance in farmers' field to obtain the status of disease infection, and encourage farmers to practice good agricultural practices, so that they will given a certificate of approval, so that the government could facilitate the farmers to sell the products in the market. And number 7 is encourage farmers to use disease-free planting material, especially from the tissue culture material. Number 8, application of crop rotation or intercropping to reduce the pest and disease husbandry. And number 9, to provide training on the disease management, especially to the extension agents and farmers. And finally, to provide service for identification, and diagnostic and identifications to for the control measures. Then we also have R&D strategy for the bacterial wilt disease management. It's a multidisciplinary approach containing breeding, ISR, biology control, and biotechnology, has been initiated under 11 Malaysian development plan from 2016 to 2020 to reviving the productions of banana, which include first, a banana germplasm screening for resistant against the disease, ISR approach, lactic bacteriophage as a biological control, identifications and validations of important virulent genes related to pathogenicity of bacterial wilt disease, DNA fingerprinting for differentiate banana accession, and finally, development of DNA-based diagnostic kit for quick and easy detections of bacterial wilt disease. The first approach is banana germplasm screening. From that, we found that 10 accession from 128 accession will highly tolerate to the bacterial wilt disease, which include pisang masam, jari buaya, IMTP, and etc., as you can see in the slide. And also 13 accession were tolerant to the banana bacterial wilt disease, including Lilin Malaysia, Berlin, IMTP 3, Otter Sarawak, and so on. This accession could be used for breeding improvement of banana in the future. Approach number 2 is ISR approach, again banana bacterial wilt disease, which involve what is actually the process for induced systemic resistance. First, to isolations of bacilli in vitro. From that, the researchers found 54 bacilli, and then they screened down up to 3 bacilli, gave total suppressions of again banana bacterial disease, if you can see in the graph. Then after that, the further screening the ISR banana seedling in the field, and from the result, we can see that the tallest banana plant with bigger leaf were treated with the bacilli coding number IB43, followed by BIB35 and IB58, shows the high performance in terms of growth. And after that, The ISA banana seedling were tested in bacterial wilt disease hotspot, where from that we found ISA banana seedling coding BIB10 have shown total suppressions of bacterial wilt disease in the hotspot, either in the first or second return as compared to others. Yeah. And for the bacteriophage, yeah, as a biological control agent, bacteriophage is a natural antibacterial entity, a virus that target bacteria. It's normally it's not affect a human or any harm to other living organism. It's a huge or abundant biological agent on Earth. Bacteria is persist in high number in any environment, as long as the host is present without any harm. Generally, phage have a narrow host range, typically being limited to strain within a particular species of bacteria. For example, Xanthomonas, Pseudomonas, Awinia, and Rastunia. The phage DNA consists of single or double-stranded DNA or RNA genome encapsulated inside a protein or lipoprotein coat. Okay, this is the process of isolation and evaluation of lactic bacteriophage in vitro in a glass house. Several flow. Yeah. First, isolation. Second, we need to screen using plug assay through double Agar overlay method. Then we have to see the visual under microscope, and then we test the bacteriophage under glasshouse environment with several treatment. If you can see there, treatment 1, treatment 10, yeah, with different application technique, drenching, injection, and so forth, yeah. And they screen the seedling using a scoring index, as you can see there, 0 to 5, and also test the bacteriophage for thermal and pH stability test. Yeah, these are the results. If you can see there, the cocktail, because they have two bacteriophage. X9105 plus X513 shows the highest infectivity effects against Rastonia syzygy subspecies salabesensis in vitro as compared with other treatment, including control. Yeah, in the glass house, the cocktail also shows the lesser percentage of disease severity at 23.3% as compared to positive control in the glass house evaluation. Yeah. The rest, it doesn't look any better than the cocktail. Yeah. So this is the thermal stability test. If you can see that the exponential formulations on plug recorded from 25 degrees Celsius, maximum at 30 degrees Celsius. Yeah, infectivity of phage decreased from 30 degrees Celsius and above, meaning that the bacteriophage couldn't survive until more than 30 degrees Celsius. Yeah, it's too the temperature is too high. And the pH stability test, the plug form from pH 6 onward and increase with increasing pH values with maximum at 10 pH. Yeah, so meaning that the bacteriophage could survive up to pH 10. Yeah. On your right side, to show you the in glass house test. Yeah. The visual symptom on the treated banana plant at 30 days after phage application, yeah, where you can see clearly that the banana plant with cocktail on the right side, yeah, treatment as a soil drenching method looks healthy, yeah, as compared to the positive control. Number 4 is identification and validation of important viral genes related to pathogenicity in banana bacterial wilt. Yeah, so this activity or project done by our biotechnology team, and they managed to get 6 different related genes from the banana plants, and including pathogenicity related protein, yeah, hypersensitive induced response protein, and so on. Yeah. So maybe this study will help to reveal virulence mechanism of bacterial wilt disease pathogen, which promise a new prospect to understand the molecular function of this pathogen and host. Do I still have time, Doctor?
Okay, you can finish it, no problem.
Yeah, I will go fast, yeah. So
Yeah.
number 5, development of ISSR marker for varietal identification, detections of monoclonal variations and breeding in banana. So the team managed to screen more than 250 banana accession to identify from MARDI itself and from neighboring countries. The banana accession vary in purity level. So 14 SSR marker developed were able to differentiate banana accession into 3 clusters. These SSR marker are essential for varietal identification, detections of monoclonal variations and breeding in banana. This is a very useful information for breeders in banana. Number 6, early detections, developments of DNA-based diagnostic kit for quick and easy detections of bacterial wilt disease of banana. So the team is also from the biotechnology. They have managed to newly developed method, a novel diagnostic method using LAMP, loop mediated isothermal amplification, incorporated with bringing fluctuations of carbon particles for binary yes or no detections of BDB pathogens. So this prototype is very useful for potential user like a government agency, especially from the Department of Agriculture, monitoring agency at the entry point, import, researcher and farm owner. So just to show you the 3 easy steps in one kit with a time about 26 minutes, and they managed to ID the bacteria banana bacterial wilt disease. So for step 1, DNA extraction just take about 5 minutes. And then application, isothermal application 20 minutes, and fluctuations assay just minute. Altogether, very short and about 26 minutes only. So, and finally, as a conclusion for my presentation this afternoon, so as a RD R&D, the base is a core business of MARDI. Therefore, as number 3, strongly support the evaluation of technologies from laboratory to the field and apps for utilizations linked to combat banana bacterial wilt disease. And number 5, sharing information like today and experiences on related issues and problems from local and foreign experts should expedite solution for the disease control. Based on the R&D, the bacteriophage cocktail isolate X9105 plus X513, none without will be formulated, and for the test in a hotspot area. And last, ISR technology is the promise the world to control banana bacterial wilt after successful in controlling papaya diabetic disease. Okay,
Okay, it's excellent work. Yeah, for the time is limited, we can only answer the 3 questions left in the chatting room. The first is that is from Tan. Is the bacteria phage that shows positive result in controlling the banana bacterial wilt will be commercialized? Okay, Dr. Rozeita.
Okay.
Is the first question.
Yeah, I will answer that. Yeah. Yeah. For the bacteriophage, at the conducted the research is further study. So by next year, she will be returned, and we hope that she managed to bacteria maybe actually local or international later. Yeah, we are very futuristic about that, so that the product can be available in the market for all.
Okay, yeah. And the second question is, which chemical is effective against BBWD? The Dr. Rozeita.
Thank you, Dr. Yun. So far there's no chemical that can cure or to reduce the infections of bacterial wilt disease. Yeah, as we know that bacteria only maybe can use antibiotic. Yeah, but in Malaysia we are not encourage growers to use antibiotic, because mostly antibiotic is focus or important to human. Yeah, for human, not really for plants. So right at the moment there's no chemical that can be used to control the banana bacterial wilt. Yeah, there's no one. I don't know, maybe in China or in Indonesia they might have, but in Malaysia we couldn't get any chemical to control the disease.
Yeah, maybe the antibiotic and the coupon products can both of them are very effective on this bacterial disease. And the third question is how to okay, there are many questions. Only we can only for the time is limited. It's a last question.
Okay.
How to have you identified the mode of delivery of bacteriophage on the field conditions? Identify the mode of delivery of bacteriophage field conditions.
The moment we only managed to do the trial in the glass house, not yet in the field. So maybe next year, if we still have the webinar like this or in other occasion, we could share the result of the mode of infection in the field condition, but not in the moment. We only managed to do the experiment in the glass house.
Okay, for the time is limited, it's time for us to enter the next session. Okay.
Okay, thank you very much.